All right, it's past, so time to continue. So Øystein, the Zoom is yours.
Yes, thanks. So let me see if I'm allowed to share my screen. Hopefully that's the case. Yeah, so I hope everyone can see this, and please let me know if you cannot. So I was asked by Otso by saying like a few words about this issue of like what else can we use HMSC for, and one of the reasons that I should say something about that is that I'm personally like quite interested in that and have been applying HMSC in some kind of exotic context, as Otso said. I just wanted to start by pointing out that I think Otso was calling HMSC something like a generalized linear something. And in this context, I want to say that it's a multivariate mixed effect Bayesian latent variable model. And the reason that I wanted to say that, I think that some like key features that we can use for a lot of things is one, that it's a multivariate mixed effect model, and second, that we have these like latent variables. And just as some like random things that makes possible is, for example, that we can estimate like extremely large correlation matrices. So by which is what essentially this omega matrix is. And what will be like my main topic over these next minutes is like what happens now if we put other things in the response matrix than, for example, species presences or species abundances. And like just some random examples from the kind of data that we have been involved in, or that even participants on this course have submitted, is like, for example, that you can have transcriptomic data where you have like gene expression at enormous amounts of genes, for example, and you want to estimate like a correlation matrix for that. And some people are working with like multiple phenological phases so that each species can enter in the response matrix many times. So what I wanted to give like as an concrete empirical example is based on a paper that Stein Joar Hægland and myself did a couple of years ago, and that came out like a year ago or something in Journal of Ecology, where we apply HMSC to try to understand what we call reproductive interactions in plant communities. And what we mean by reproductive interaction, these are like interactions that are about attracting pollinators, basically. So instead of species interactions in terms of like, for example, competing for nutrients, we are here interested in things like whether two species share the same pollinator and whether that affects the fitness of the species. So if you consider like this meadow in the background, for example, we can ask, okay, does it matter for this yellow species that it's surrounded by this blue species? So in this paper, we basically outlined several examples of like data types that we can think about for these kind of questions, and where we can use HMSC to analyze it, and we try to give like concrete examples of how to use HMSC. So for each of those, we try to show kind of what goes in the response matrix, what goes in the covariate matrix, and so on, and also which kind of outputs that we can use to interpret. And the example that I will go through in some detail, we consider a situation where in different plots, you have different combination of plant species, and we are interested in how much they are visited by pollinators. And that means that like in the Y matrix in HMSC, instead of like the occurrence of the species or the abundance of the species, we put now, for example, the number of pollinator visits that each species received during, for example, some observation periods. Then in this case, as covariate, we actually put exactly those same species, but instead now of the number of visits that the species received, we put, for example, the number of flowers that the species has there. And what we will do with those data is we will ask, is it so that, for example, the red species is visited less in those plots where the blue species is more abundant? So in this data, we had something like 9 species occurring in 20 plots, and my collaborators collected like 200 observation censuses where they went to the plot and they counted like how many pollinators, in this case bumblebees, went to each of the species. And to analyze this, in this case, for example, we chose to take like a stepwise approach. So we started by fitting a model where we completely ignore like all these flower abundances and so on. So the only covariate we had was something like temperature, because we think that is like important for pollinator activity. But otherwise we kept only random variables. So we put in there like the plot and then the particular observation unit. And this allowed us then to generate what we have been calling in the course raw associations. So these are basically patterns of correlation, assuming like no influence of those covariates that we are actually interested in, but we want to know first, okay, what are like the associations in the data? And in this case, there were like some of them suggesting that in cases of positive association, the two species tend to be visited at the same plots. And in terms of a negative association, it means that the species tend to be visited in different plots. But now, of course, this is like an overly simple model of this system. And first, it's so that naturally it will be so that if a species is not present in a plot, it will not be visited. But if it's like very common, it might be visited a lot. So then we built a model where we include for each species, like how common was that species. So now we are talking about purely within species effects. And we can think about that as a kind of interaction matrix, which is now still a square matrix, just as we have been looking at before. But notice that in this case, the parameters estimated here, they're actually the fixed effect in the model. This is not like this omega matrix, but it's just for each species. What is the effect of your own abundance on how much you get pollinated? And unsurprisingly, that is always a positive effect, although the strength differ a bit among species. Then to really get the species interactions here, what we wanted to do was, of course, now to look at what is the influence of all these other species. And that has kind of two parts, but let's focus now on like what we have put in the fixed part of the Hmsc model, which is that we now put as predictors of the visitation to each of the species, we put the abundances of all the species. In this case, we did not use any like reduced rank regression or so, but because we had only 9 species, we put all of them. But of course, here we could have also used things like reduced rank regression if we had, let's say, 100 species. And how did this look? Then it looked something like this. So we get a complete matrix where we have like some positive effect and some negative effects. And in this matrix, what we are looking at is like, what is the effect of the abundance on the species on the x-axis on visitation to the species on the y-axis? And what's important to note here is that this matrix is no longer symmetrical, like the omega matrix is, for example, but this one is actually directional. So that, for example, we can have an effect that is positive in both directions, suggesting that maybe these species together are attracting pollinators, and then when they together do that, they are both visited. We can have effects that go in only one direction. We can have effects that go in opposite directions even. And of course, those become a little bit more complicated to interpret, but the important thing is that by having this kind of model where the interacting species are in the fixed part of the model, we can actually get a matrix that has some direction to it. Then I will not go much more in detail. I just wanted to mention briefly some other ideas we had about types of data that can be analyzed. So for example, it's often so in pollination biology that people have a plant species they are interested in, and they go out and record which pollinator visits it. And very often that's more than one pollinator. So it can be, in this case, it can be some birds, and it can be some butterflies, and it can be some bees, and so on. And then of course we can use Hmsc to analyze such data and see, for example, if there is association, so that, for example, birds and butterflies tend to visit different flowers and so on. we can think about like effect of species traits. So in this case, we are talking like about phenotypic traits of individuals. So that in this case, we put actually phenotypic traits in the X matrix and ask how, say, the size of the flowers and the height of the plant affect the pollinator visitation. And then in addition to that, we can, for example, look still at residual associations, which would suggest, is it so that, for example, after we have control for all these traits that we think are important, is it so that still the species tend, for example, to be visited more together, suggesting some kind of facilitative effect, or that they are not visited together, suggesting a competitive effect? So basically just as in occurrence analysis, but now focusing like on any kind of fitness measure such as seed set, or it could also be fitness components such as pollinator visitation. And then finally, I just wanted to mention something that I so far haven't tried, but I like every time I hear a talk about networks, which lots of people work on in pollination, I like think that this would be like a cool thing to use Hmsc for. So now we imagine this matrix where we have, for example, plant species and pollinator species. And then it's of interest to know, can we predict the structure of these networks by considering the phenotypic traits of the plants and phenotypic traits of the pollinators? And this I think we can quite directly do with Hmsc by putting, for example, the traits of the plants in the X matrix and then the traits of the pollinators in the trait matrix, and looking, for example, at these R-square and trait R-square values to quantify these effects. Yeah, and with that, I think I will stop, and I can see the chat is blinking. So just in case that's all questions for me, I will stop there, and we will take questions and then go on to the general discussion. Thank you.
So the comments in the chat haven't been directly about the presentation, unless something has just popped up. Okay, it's been a discussion about possibly similarities between Hmsc and other methods that take multiple species and produce distribution models or abundance models. If someone wants to ask a specific question about okay, now we do. Is there a way to include co-phylogenetic signal, do you think?
Right, so in the terms of like, so co-phylogenetic signal, I tend to think about things when you have like two phylogenies, so like the phylogeny of the plants and the phylogeny of the pollinators and so on. And I haven't thought about like that in particular here, but what I can say as a general note that in this like plant pollinator world, it very often turns out that actually there isn't so much of that. It's more like that plants are adapting to pollinators, but generally we don't find very much that the pollinators are adapting to the plants. But it's worth thinking about. And for that, and in general, if any of the people here are working on any data like this and are interested in discussing or collaborating or so on, I would be happy to hear about that.
Can you see the chat yourself? There's a question now.
Right, so now there's lots of things. Some are private to me and some are not.
Oh.
So I will just take some random ones here. So Eleanor is asking, how would you suggest one include taxa in one analysis that one is not as clearly sure of their linkages? So not that sure I fully understand this, but like of course in pollination biology, we are quite sure that it's like the insects that are pollinating the plants and not the other way and so on. But I think if you mean like more generally that you have species that you wonder if they are interacting, but you don't know like exactly how, then I think in general, putting everything in the Y matrix and looking at these association matrices is probably the best start. And Maria is asking, how did you arrive at the directional association as opposed to symmetrical? And that was because in this case, what I presented was like the beta coefficients rather than the residual correlations. So a bit similar to Otso's time series model. Okay, but I think like now it's more or less time to go on to these general issues, because as I said, we have.
Sorry, one more specific question, which I thought was interesting, was a question as to could you dig into something like selection gradients based on the results?
Yeah, so that was actually, I chose not to say it because it was a little bit complex to explain, but actually my second example where I let like the phenotypic traits of individuals vary. Actually, the point of that analysis was to estimate selection gradients. So in the supplementary materials of the Journal of Ecology paper, there is like some simulated data on how you can use Hmsc to estimate selection gradient on multiple species simultaneously. And that's something that I'm quite interested in trying out more, because it kind of gets at this, how does the presence of potentially interacting species affect selection on a focal species, which I think is like a largely unexplored issues, at least in the pollination world.
Okay, and there's more follow-up now. So people are just coming up with their questions. So another one was, okay, that we enjoyed hearing about the intraspecific interactions, but it would have been, if I'm understanding this right, nice to see more about spatial overlap in occurrences. So the question was, could it have been solved by, I guess.
So overlapping occurrence in a spatial, so you might then be thinking about like things with how, like, for example, can we, let's say that we had a lot of data on the distribution of plants and distributions of potential pollinators. Could we then use like those data to, for example, find out candidate plant pollinator interactions? And that's something that we have somehow been trying both with those kind of data and like with butterfly host plant data and so on, but very often it ends up in the same problem of how do you separate like joint environmental responses from actual interactions. So I think at least then having some kind of field observations or field data to link it to would be very important to make sense of that.
And also there was a question about how do you account for potential biases within or among observation plots? I don't know if is that observer bias or what kind of bias do you mean, J. Leon?
Well, I mean, of course, in any science, there is any kind of bias we can worry about. But
okay, observer bias. So, do you mean by the person observing the butterflies? So, or just
I mean, like I can only answer like very generally about that, but like of course, just as when using Hmsc to model species distributions, of course, we can put things like observer effort in there. We could also put, I guess, like the name of the observer to assess if that explains any variance. And then hopefully saying that the observer explained like 2% of the variance, and hence we can ignore it.
Okay, thanks very much, Øystein. I think this was a really nice presentation to show a bit non-standard use of Hmsc. As so far we have really focused on just the standard output, and that's of course only the starting point. I saw that in the chat there were some questions about comparisons between Hmsc and some other related approaches. Those were already partially answered, but I wanted to share a couple of slides related to this, and this relates to this theme of what does actually this joint mean in the joint species distribution model. And now, as I have tried to explain, at least in Hmsc, it's a joint species distribution model, not a stacked species distribution model, for two reasons. One is that it has this niche structure, So it's doing some joint modeling of those beta parameters, those species niches. It can look at their dependency on traits and phylogeny, and also without any traits or phylogeny, it's still estimating them jointly by assuming that they have a common mean and some variance covariance matrix around them. But then the other reason why a joint species distribution model is a joint model is that it looks at the biotic interactions, or more correctly, the residual associations. And this is something that you cannot look at if you just model the species one by one, because you look at how correlated the species are in the residuals at various levels. And here I have pulled out a couple of other kind of a major joint species distribution packages on top of Hmsc, such as Boral, JCHAM, GD, SDM, NISTNet, SAM, the species archetype model, BC, so the Bayesian community analysis, and SSDM, which stands here for stacked species distribution models. So pick your favorite single species model and just apply it one by one to each species. And then I try to understand which aspects of these joint species distribution modeling each of these have. And I apologize if I missed something. I haven't followed up the development of each of these packages, like what they do right now, but this was, in my understanding, the situation when I prepared this slide. So some of actually all of these packages, maybe except SAM at that time, estimated the species association networks. So a lot of these joint species distribution modeling has focused on the species associations. Most of these models also have some kind of a niche structure, so that they try to estimate those betas together, that there is some hierarchical prior or something like that for those. Much less, in my understanding, try to bring in the information about the traits or the phylogenies when modeling the species niches. I'm not saying that none of these other approaches do it at the moment. Maybe they do now, but this was the situation some time ago. But more interestingly, in my understanding, none of these approaches, including Hmsc, is modeling the species associations as a function of species traits or phylogenies. This is one development need that we see for Hmsc. The reason why we haven't done it so far is that it's a bit tricky. It's more difficult to do. It's difficult, maybe. What is difficult is to write down a nice equation that models how species associations depend on traits and phylogenies. Because you could think that, okay, let's just do the same regression model that we do for the beta parameters, that now the lambda parameters, they are analogous to beta parameters, that let's model them as a function of species traits and phylogenies. This could be implemented, but actually it leads to a situation where similar species in terms of traits or phylogenies, they can only have more similar responses, or they can only have positive associations, not especially negative associations, just because of the mathematical structure of this model. And quite often you would like to have similar species to have negative associations, because they are the ones that, for example, are expected to compete with each other more heavily. So at least I don't know how to do this yet, but hopefully somebody will figure it out at some point and fill in this gap here. So this is one way to compare joint species distribution models, what kind of a joint structure they try to estimate, and then a related way to compare these and stacked species distribution models. They do none of this because they are not joint models. And then another way to compare them is what kind of data can be utilized by those joint species distribution models. Whether they can utilize the community data, yes, all of them can do. Whether they can utilize the environmental predictors, yes, all can do. But then less approaches, at least when I've compiled this table, could utilize trait data, phylogenetic data, or do the models in a spatiotemporal context. And then a way in which you can compare these models is in terms of their predictive power, and that's what we did in this paper by Anna Nurberg and others. from last year in ecological monographs, where we fitted a bunch of models, both joint species distribution models and stacked species distribution models, to various data sets, like real data. They were not simulated data, but real data, and then ask how well these models do in extrapolation and interpolation, and then we evaluated the predicted performance in many ways. So this is just a summary where we kind of sum the scores and look at the overall predictive power of these approaches. And actually what you see in this figure is not so that the joint species distribution models all do similarly. They do very differently. So what you have between these joint species distribution models are all kinds of single species models, for example, random forests and GAMs and all of that. So it's not so that the joint models do well in this kind of a case and the individual models do well in this kind of a case. It really depends more deeply on the model structure and all kinds of small but important technical choices that you have to do when you implement the models. And of course also on the type of the data, that in this comparison, the Hmsc, or more specifically the Hmsc with spatially structured latent variables, was the one that did the best on average, but then in some other data sets, some of these other ones might do much better. So here this was one comparison, and one take-home message from this is that there are many joint species distribution models, and they are, I think this is like a very exciting field in the sense that there is a lot of development here by many groups all over the world. And so the field is developing fast, which is great. Better models will be available in the near future, but it also makes it maybe difficult to apply these methods, because there is maybe some confusion about what is the set of methods available. It's not like well established yet, like what are the standard methods, and also because all of these are developing all the time, like we are implementing new features to Hmsc and so on. So it's more tricky to apply them because they are not yet, you cannot look from a textbook 5 years ago that how does this actually go.